We just finished the first podcast on population genetics. In this podcast, we're going to continue our discussion with population genetics and focus on the Hardy-Weinberg equilibrium and the equation that we use to predict genotypes in the next population. All right, let's go ahead and get started. Okay, so now we have just learned how to predict allele and genotype frequencies within a population. And that gives us a better grasp of the different kinds of genotypes, the different kinds of phenotypes within a population, and that's useful information. But can we use that information to make a prediction about how stable, genetically speaking, that population is? For instance, if you saw in one population that the allele frequency of big A was 30%, is it going to stay 30% in the next generation and then the next generation? How stable is it genetically? And that question was pondered by, the first was a British mathematician by the name of Hardy, and the other was a physician from Germany, and his name was Weinberg. The two individuals never worked together. I'm not sure if they ever met, but they came up with this concept and this equation that we're going to talk about independently to explain how we might be able to predict what the next generation will look like if we know what the allele frequencies are in this generation. So the Hardy-Weinberg equation helps us predict the allele and genotype frequencies in the next generation when certain conditions are met. So I will talk more about these conditions and what they mean, but while it's on our mind, I want to at least write down what the Hardy-Weinberg equation is, because we'll come back to this. It's p squared plus 2pq plus q squared equals 1. P squared equals the homozygous dominance. PQ represents the heterozygotes, and q squared represents the homozygous recessives. So if you draw a little Punnett square up here, and let's just draw big A, little a, big A, little a, that's all we're talking about here. Big A, that's our p squared. The frequency of big A in a population times the frequency of big A in a population, p squared. And then we have big A, little a, big A, little a. Again, we think about the population, the frequency of big A, little a in a population, and how we might be able to predict the number of heterozygotes we'll see in the population. And so it's P times Q, and since there's two different ways to get that, we multiply that by 2. So this is really big A, little a in this example here. Q squared is our homozygous recessives. The probability of getting this genotype, little a, say, in this example, by knowing what the frequency of little a is in the population. And it's always going to equal to 1. Add up all the different alleles in a population, it should equal 1. But the only way we can make this prediction is if these certain conditions that I vaguely mentioned here are met. In fact, we know that if these certain conditions are met, that the frequency of alleles and genotypes should not change in that population. But let's talk about what these conditions are before we move forward. There are 5 conditions. Sometimes we call these assumptions. So if I slip and say assumptions, conditions and assumptions are the same thing in this case. The first is that the population is large. And what this does is it prevents changes due to genetic drift. Meaning, if you have a population of 1 million individual ladybugs, say, and you lose 200 of them because of some sort of epidemic, maybe a kid stepped on them, something like that, the overall population doesn't change much because it's such a large population that these random fluctuations in the population aren't going to affect it. Random matings. And this means that the gene that we're studying doesn't confer any mating advantage. Every member in that population has an equal chance of mating. No new mutations. So no new mutations in the gene we're studying. That's a really important point that I glossed over earlier, but I should stress it right now. And that is that when we're thinking about these conditions of the Hardy-Weinberg principle, and we're making the predictions about whether the allele frequency will change, all we're doing is looking at one gene and its associated alleles. This principle only makes a prediction about one gene pair at a time. So no new mutations in the gene of interest. 4. No migration. So no migration means that we are not adding to the gene pool. So the gene pool is not changing. And the last one is no natural selection, meaning that the gene we're studying and that's associated alleles does not confer any reproductive advantage. So all the members have the same reproductive success. If one member of the population was able to pass its genes on more effectively than the other members of the population, then we would see a change in allele frequencies. But as long as all 5 of these conditions are met, we should not see any change in allele frequency. Okay, I've mentioned it a few times, but let's go ahead and put it in writing to make it official. If those 5 conditions, so if 5 conditions are met, then we know that the allele frequency of the gene of interest will not change in the next generation. Other thing we know is that after one generation of random mating, the genotype frequency is stable. Another way to think about this is we can say that if these conditions are met, then the allele frequency alone, no outside factors, only the allele frequency determines the genotype frequency. If we know how many alleles we have in a population and all these conditions are met, then that is all that is needed to be able to make that prediction of what the genotype frequency will be. Now we can list a fourth one here, and we can say that if the 5 conditions are met, the population is in Hardy-Weinberg, HW abbreviated, equilibrium, EQ. And we know that a population will stay in this equilibrium as long as these conditions are met. But as soon as one of those conditions are not met, it falls out of Hardy-Weinberg equilibrium. So if there's natural selection, if there's migration, if there's non-random mating anymore, then the population will fall out of Hardy-Weinberg equilibrium. Now remember, when we're looking at this Hardy-Weinberg equilibrium is only measuring one gene and its associated alleles. And in general, most populations, particularly if you look at all of the genes in a population, very few populations would ever be in Hardy-Weinberg equilibrium. So I'm just going to write Hardy-Weinberg equilibrium in the whole population, looking at many genes, is rare. Well, if that's the case, then what use is this equation and thinking about a population in or out of Hardy-Weinberg equilibrium? Well, what the equation helps us do is it helps us explain evolutionary forces. So it helps identify evolutionary forces. These 5 conditions are examples of an evolutionary force, something that can change the allele frequency within that population, and that can sometimes drive evolutionary changes. Next, I would like to list some consequences associated with a population in Hardy-Weinberg HW equilibrium, EQ. The first is, as long as this population is in Hardy-Weinberg equilibrium at that gene that we're studying, there is no evolution with that gene. It would probably be better to say there's no evolution within that population in regards to that gene, because that gene and its frequency of the alleles don't change, so it's not going to affect the population at all. We can determine genotype frequencies by allelic frequencies. And the best way to look at it is to look at this little figure here. And you can see in this figure here that if we know the frequency of, say, big A and the frequency of little a, we can make a lot of predictions about the genotype. So for instance, if the allele frequencies for big A and little a are between 0.4 and 0.6, they have to add up to 1 ultimately, but if each of them is between 0.4 and 0.6, then we know within that population we're going to have more heterozygotes. If, for instance, say big A, we have a smaller population or smaller frequency of big A and a larger frequency of little a, then what we'll see is a larger percentage of individuals in that population who are homozygous recessive. Of course, the opposite is true, and that is if we go and have a higher frequency of big A versus the frequency of little a, then we'll see more individuals in that population who are big A, and then all the different frequencies in between. But using a table like this, as long as the population is in Hardy-Weinberg equilibrium, we can make a lot of really good predictions about the number of heterozygotes or homozygotes within a population. Another important question, and I love to ask questions similar to this on tests or quizzes, and that is that dominant traits are not necessarily high in frequency. So if we look at our graph again, big A is the dominant trait, but if there happens to be more little a's in the population, then the number of individuals who have the dominant phenotype, big A, are going to be smaller. So just because big A is dominant does not mean that more people in that population or more individuals in that population will have the phenotype of big A. It could be they have the phenotype of little a. Often we see the word dominant, and in our minds that confuses us, confuses me sometimes, and we sometimes think that that means it has to be the most common. Dominant and most common are not the same thing. And this last thing is one of the more important things I would imagine, and that is that genetic variation can be maintained. Within any given population, usually, you can find some examples where this isn't the case, but in most cases, having a lot of genetic diversity is going to be important to maintain that population. And as long as it's in Hardy-Weinberg equilibrium, you can maintain that variation. Alright, so let's walk through this example of how we can use the Hardy-Weinberg equilibrium to make some predictions. So let's say in this gene pool that we're looking at, that the frequency of big A is equal to 0.7, and that the frequency of little a is equal to 0.3. They always have to add up to 1. And so what this means is that within this population, we can be assured that 70% of the gametes that are made will have big A alleles in them, and 30% of the gametes will have little a allele in them. So let's take a quick jump back in the past, and if you remember back in the day, last week that is, when we did these Punnett squares, and we went big A times little a times big A times little a, and we came up with something that looked like this. And this made sense, because we were talking about an individual. And within an individual, when we're crossing two individuals, within a population or not in a population, doesn't really matter, this is what we would expect their progeny to look like. But we want to take it that one next step, that step beyond that individual and their progeny. But how does the frequency of these alleles affect the whole population? So instead of doing an individual Punnett square like this, let's change it a little. Alright, so let's look at how we're going to do this pedigree now. Instead of saying big A here, we're going to say the frequency of big A, and then the frequency of little a. We could also call this P, and we could also call this one Q. Over here we're going to have the frequency of the population of big A. We could also call that P, and then the frequency in the population of little a, and we could call that Q. Now let's bring them together. And what we get is the frequency of big A, the frequency of big A, little a, and the frequency of big A, little a, and the frequency of little a, little a. So essentially what we're looking at here is, this is P squared, this is PQ, PQ, and Q squared. Frequency of big A and big A, so that's A times A, or we might call it P squared. And remember, that is what our Hardy-Weinberg equilibrium was. P squared is the probability of having a homozygous dominant. 2PQ is the probability of having a heterozygote, where you get the big A from one parent, little a from the other. plus the probability of having the reverse, where you get big A from one parent and little a from the other parent. So that's why we go 2PQ. So there are two different ways you can be big A, little a in this example. And then the probability of being little a, which is Q squared. And this always has to equal 1. So we know a little bit more here, because we know what the frequencies of big A and little a are in this population. So we can actually solve this. Because we know frequency of A is 0.7, so this is 0.7 times 0.7. Or over here, 0.7 squared is 0.49. Big A times little a is 0.21, because it's going to be 0.7 times 0.3. Frequency of A times the frequency of little a. And it's going to be the same here. And over here it's going to be 0.3 times 0.3. Right, so let's go ahead and calculate this. 2 times P times Q, so it's 2 times 0.7 times 0.3, 0.42. And then Q squared is 0.3 times 0.3. And that's going to be equal to 0.09. If you add all that together, you'll get 1. So we know that as long as the population is in Hardy-Weinberg equilibrium, that the frequency of big A will remain 0.7 and the frequency of little a in that population will remain 0.3. If we know that, we can plug it into this Punnett square, or just into this equation, that the frequency of homozygous dominant individuals, big A, in the next generation will be 0.49, or you could say 49%. Proportion of heterozygotes will be 40.42 or 42%, and of homozygous recessives we'll say 0.09 or 9%. Again, by knowing these frequencies, we can make a prediction about the next generation if those 5 conditions are met. So what happens when one of those conditions are not met? Well, if they're not met, we know it falls out of Hardy-Weinberg equilibrium, and whichever condition is not met will help explain some of the evolutionary drive within that population and how the genetic variation will change, either increase or decrease depending on the situation. But I want to talk about one of the situations when it's not met, and that is random matings. And to do this, I want to talk about a scenario, and the scenario includes the bighorn sheep. You may have heard of these as just by the name of rams. Their population at one time, say around 1900, was greater than 2 million. As we started to settle out west more, and more people went out west, that number plummeted to 70,000 bighorn sheep. In 1922, they placed 12 lucky bighorns on a national reserve where they were protected. They had plenty of habitat, they weren't going to be hunted, they were safe there. Within 8 years, so by 1930 roughly, these 12 had grown to 90 bighorn sheep. So it was looking good for this population. Over the next 50 or so years, their numbers started to gradually climb and get more and more and more. However, they started to drop at one point. And by 1985, their numbers had dropped to 50 bighorn sheep. Let's go back here and look at our original 2 million sheep. When this population was thriving out west, they met all the conditions. It was a large population, random matings, no natural selection. But when people came out there, their numbers started to drop. And they fell out of Hardy-Weinberg equilibrium. And that's why their numbers dropped to 70,000 and continued to drop. Now when we placed 12 of them within this national reserve, they started to grow again and get more and more in numbers. Once again, they were almost in Hardy-Weinberg equilibrium. The problem is their numbers were still pretty small. So they were susceptible to things like genetic drift, where if something happened to these 5 of them, that would cause a dramatic problem within this population. And so even though this worked out pretty well for a while, the problem is that these 12 bighorn sheep, that was their gene pool. As they grew to as numbers as 90s or to a few hundred. That gene pool came from these original 12. And since you only had these 12 here, there was a lot of inbreeding within this group here. So when they had progeny, those progeny would end up having progeny with other related bighorn sheep, either possibly siblings or possibly what we might call first cousins within the sheep world. And so what they did here was that they had to find a way to rebuild the gene pool or add to the gene pool. So what they have done is every few years they add about 5 male bighorn sheep. And just adding this little bit of genetic diversity back into this reserve, that has been able to stabilize the population. So every so many years they just add a few more male bighorn sheep. And that seems to have done the trick. And we call this, when we see a population that has a reduced gene pool, and we add more to the population, we call that a genetic rescue. So with that as our backdrop, let's talk about the problems of having non-random matings. This, of course, is showing one example of it where you had these 12 sheep that were inbred, and the progeny were then further inbred. And let's talk about these two terms called positive assortative matings and then negative assortative matings. The positive assortative matings is when matings occur between like individuals. Negative assortative matings means that matings occur between unlike individuals. They're still the same species, but they differ in certain traits. For instance, maybe here you have a taller breed of ram mating with a taller breed of ram, and maybe over here it's a taller mating with a shorter. Something like that. Sometimes within a population you may have examples of both of these. Maybe with one trait the matings show a positive assortative pattern, whereas in other traits within that population it's negative. And as with the bighorn sheep, we saw inbreeding. And inbreeding is a perfect example of positive assortative mating. So let's erase this. And I want to talk about something known as the inbreeding coefficient. As we describe inbreeding coefficient, let's go back and look at our bighorn sheep. So as we talk about inbreeding coefficient, let's look at these two genes, big A and little a. Big A confers a healthy respiratory system. Little a confers a disease respiratory system. Mainly, they're much more susceptible at catching pneumonia and various other infections. It's a recessive allele, so as long as you're big A and big A or big A little a, you're healthy. Or rather, the bighorn sheep is healthy. But if you're little a, less healthy. So the idea is to keep the numbers of sheep either big A, or heterozygotes. We want to make sure, if you're interested in this population, which we are, we want to make sure the homozygotes stay low. And we see when we have inbreeding, we have an increase in homozygotes. And that's what I'd like to try to explain to you. And over here I'm going to draw this, and finish drawing this graph here. And along the vertical axis we have homozygotes in population. Along the horizontal axis we have the generation of breeding. So we'll just put generation. And we know an individual who is in a random mating population that the number of homozygotes will remain relatively low. And we would say they have a coefficient of inbreeding, which we call just F, which is equal to 0. Don't worry about how we calculate it, it's just 0. You should remember that part though. And we know if the bighorn sheep were to mate with a bighorn sheep that was, say, a first cousin of it, its coefficient for inbreeding would be 0.0625. And then, if two bighorn sheep in this population were to breed with siblings, then its coefficient for inbreeding would be 0.25. Now this wouldn't happen in bighorn sheep, this next example I'm going to draw for you, but it does happen in some organisms, and that would be the self-fertilizing organisms. And in which case their coefficient for inbreeding would be 1. Again, this can't happen with bighorn sheep because they cannot self-fertilize. But some organisms can. So how can we use these numbers? Alright, now let's look at how we're going to use these numbers. Ultimately, you want to know what the probability is in your population that you're looking at that individuals will be little a, little a. And if the frequency of the A allele is equal to 0.01, say, and we want to know what the probability is that in a population that is in Hardy-Weinberg equilibrium, so there will be random matings, so less inbreeding, we know that the coefficient for inbreeding will be equal to 0. I should label these lines up here. As I'm looking at it, I didn't do that. So let's just say, remember 0 is Hardy-Weinberg. This one was first cousins. This one is siblings. And up here on top was self-fertilizers. You probably wrote them down while I was saying them, but I wouldn't have slept well tonight if I knew I didn't write those down. Alright, so let's come back to our problem. We want to know what the probability in this population that we will have homozygous recessives. And what you do is you solve this equation. Q squared plus PQ, the probability of a heterozygote, and you multiply that by your coefficient of inbreeding, which is F. So in this example, the probability that we would expect a homozygous recessive individual, a little a in this case, would be 0.01 times 0.01, which is equal to 0.0001. Or you might look at this and say, that's 1 in 10,000 sheep. Pretty rare. Alright, so now let's look at our sheep in the reserve. Remember, in this situation, there is inbreeding going on because we only put 12 sheep there. And let's look at the more extreme example of sibling inbreeding, where the coefficient is 0.25. We're still going to solve the same equation, except up here, because the coefficient was 0, we only had to consider this, because this cancels out. So again, we're going to go Q squared plus our coefficient for inbreeding, which in this case is now 0.25, and then we're going to multiply that by our frequency of big A, which is 0.99, and then we're going to multiply that by our frequency of little a, which is 0.01. Again, our frequency of little a, as you can see, is not very high. It's still pretty low. But because of the inbreeding with siblings in the sheep population, we're going to see that number go way up, the probability of getting little a, little a. So let's go ahead and plug these numbers into our calculator. And what you should get is 0.0026. And even though this number looks fairly low, it turns out to be quite a bit higher than what we had before. This is 26 times the amount than we previously had. So that's a dramatic increase. So let's say this again briefly. When the population is in Hardy-Weinberg equilibrium, and we know the frequency of a mutant allele like little a, we can predict the percentage of individuals in that population who will be homozygous recessive. You can also predict when the population falls out of Hardy-Weinberg equilibrium due to inbreeding. And using our coefficient of inbreeding numbers here, we can calculate what the new probability is that an individual within that population will have this disease that makes them more susceptible to respiratory infections. This is something we call inbreeding depression. And this is one of the reasons the 12 sheep that originally were placed on the reserve, if there happened to be a mutation in them, which there was for this little a, over time you had more and more sheep with a little a in them, a little a allele. And that gradually grew over time until you had a weaker overall population. And remember, they solved this by introducing new male bighorns to the population. So by using the Hardy-Weinberg equilibrium, we identified one of the forces of evolution within this population, and that force was having non-random matings, or more specifically, having inbreeding within that population of bighorn sheep. As we close this podcast on population genetics, I want to spend a little time talking about humans. And we've spent this podcast talking about how allele frequencies can change or remain the same within a population. And when they change, it's because they fall out of Hardy-Weinberg equilibrium. And we discussed those 5 conditions that allow that to happen. And when they fall out, then we can get a change in allele frequencies. And those changes in allele frequencies within that population may allow for advantageous adaptations for that population, which allows them to continue to evolve. And so humans, if we start way down here where hominids are, a common ancestor for all primates, hominoids I should say, we know that the gorillas came up here, and we know a branch came up here that would become the chimps and the bonobos, probably my favorite primate, well maybe outside of humans, maybe. And then we had a branch that came up here, and we had our Neanderthals come off here, and they coexisted with Cro-Magnon, and Cro-Magnon eventually was an ancestor to us. We end up with Homo sapiens. We know we can look at genetic differences between Homo sapiens, bonobos, chimps, and gorillas, and we can look at some of the genetic variations that we see in fossils from Neanderthals and Cro-Magnon, and we can see some of the changes that have occurred genetically. And some of these genetic changes have provided certain adaptations that have allowed chimps to survive within the selective pressures that they faced going up the evolutionary tree, and same with bonobos and gorillas. And of course same with Homo sapiens. So what are some of the genetic mutations that have occurred to allow Homo sapiens to have survived and adapted to our environment? And so I'm going to erase this here, and I'm just going to list a few. We could spend an entire course talking about these. The keratin gene. This affects hair coverage. This particular allele is expressed in chimps and gorillas and bonobos. So they express this keratin gene. Humans have a nonsense mutation in this gene that is expressed in chimps and gorillas. So we don't express it. We have less hair covering. The FOXP2, which we've talked about before this semester. And just to remind you, the difference between chimps and humans is only 2 amino acids. So not much at all. And we also talked about how it's found in mice, finches, and so forth. Hemoglobin genes. We're just going to put HB for abbreviation. As humans have evolved, we have gone through gene duplications, as we've talked about before. And that gave us different forms of hemoglobin. Remember we talked about we had a fetal form, we had an embryonic form of it. And this has allowed for a longer fetal period and for an increased brain growth. So this has been very advantageous for humans. Genome expression, just as a general feature. When you compare humans and chimpanzees, we know that genome expression in the brain is increased in humans. We have more genes being turned on. We have more transcription factors in the brain. We also know if we look at the liver between humans and chimpanzees, that the difference in genome expression is not that much. So our livers are pretty much doing the same job. But our brains have remarkably changed compared to the chimpanzees. I don't have room down here, so I'm going to put it up here as our last point here. Humans have more genes for organ development, which has also impacted our ability to adapt and evolve. But this is all I have for this podcast. In this podcast, we've talked about many different things. We talked about, in a brief summary here, we talked about calculating allele and genotype frequencies. We talked about the differences between polymorphic versus monomorphic. We talked about different ways that genes can be mutated that can change our allele frequencies. We spent a lot of time talking about our Hardy-Weinberg equilibrium and the equation. We talked about what it meant to be in Hardy-Weinberg equilibrium, and we talked about those 5 conditions, which we may have mentioned once or twice in this podcast. So those 5 conditions that hold a population in equilibrium, and when one of those breaks down, the population falls out of equilibrium and is allowed to evolve. We spent some time talking about one of those conditions when it is no longer held in check, and that was the random mating, and how when you don't have random mating, how that can lead to inbreeding, and how the inbreeding in that sheep population that we talked about as an example can lead to an increase in homozygous recessive sheep, or individuals, I'll say. As I've said a few times in class, most organisms, humans included, are walking around with a lot of deleterious mutations, but they're protected because they exist as a heterozygote. But if we increase the probability of having these homozygous recessives, then we increase the chances of individuals having these rare conditions. And finally, we closed with talking a little bit about human evolution. Alright, well, that is all I have for this podcast. And sadly enough, it is all I have for this semester. This is our last podcast. It's been a good semester. We'll see you around. Bye.